The chemical senses are your sense of olfaction and gustation, or your sense of smell and taste. Chemoreceptors are found in the olfactory epithelium for your sense of smell and in the taste buds for your sense of taste. These two senses are very closely associated. The olfactory epithelium is a pseudostratified epithelium that's found in the roof of the nasal cavity. This is really a pretty poor location for a very acute sense of smell. That is why if we sniff, we increase the stimulation of the olfactory epithelium. The olfactory receptors are bipolar neurons. The olfactory epithelium is in the roof of the nasal cavity. The olfactory receptors are bipolar neurons that are in the roof of the nasal cavity. As air comes in, it will go across this olfactory epithelium to stimulate these cells. The olfactory bulb is made up of the secondary neurons that carry the information into the central nervous system. The olfactory bipolar neurons are surrounded by supporting cells and olfactory stem cells. The olfactory stem cells allow for regeneration of the bipolar neurons every 30 to 60 days. Each of the bipolar neurons has a thin dendrite that terminates into a knob that has protruding cilia. This increases the surface area of the dendrite. The supporting cells produce mucus. The mucus covers the cilia, and that is where the chemicals become dissolved so they can come in contact with the receptors. The bipolar neurons are the receptors. Here are the cilia on them, the mucus that covers them so that we have a fluid that will dissolve the odors. These will synapse in the olfactory bulb with the mitral cells. Humans can distinguish about 10,000 different odors. It is approximated that we have about 400 smell genes that are active only in the nose. Each one of these genes encodes a unique receptor protein. Each protein responds to one or more odors. Each odor can bind to several different receptors, so odors are combinations of different receptors. Each receptor has one type of receptor protein. Pain and temperature receptors are also located in the nasal cavities. In order for us to detect an odor, the chemical must dissolve in the mucus of the epithelium and bind to the olfactory neuron. This will cause depolarization of the olfactory neuron. The bipolar neurons, which are the receptors, will synapse with mitral cells and the olfactory bulbs. These are the second-order neurons that carry the information into the central nervous system. Besides relaying the signal, the mitral cells also refine and amplify the signal. The information will travel via the olfactory tracts through the thalamus to the olfactory cortex and frontal lobes. This will help us identify the odor. The hypothalamus and the limbic region will add emotional response to odors. Odors are one of the strongest evokers of memory. The receptor organs for taste are taste buds. Most of the 10,000 taste buds are on the tongue papilla, those little projections on the surface of your tongue. There are a very few on the soft palate, the cheeks, the pharynx, and the epiglottis, but these do not significantly contribute to our sense of taste. Taste buds are composed of supporting cells, the gustatory receptor cells, and the gustatory hairs that project through the taste pore on the taste bud. Sensory dendrites wrap around the taste bud. Taste buds can be replaced about every 10 days. Most of the taste buds are on the papilla of the tongue, and we have different kinds of papilla. The taste buds are down in these depressions in the tongue. We have the gustatory cells, the supporting cells. Here are the fibers of the neuron that go to the gustatory cells, and the gustatory cells will have the little hairs that project through the taste pore. This is the part of the receptor that will pick up the chemicals dissolved in saliva. We have 5 basic taste sensations: sweet, salty, sour, bitter, and umami. Umami is Japanese for savory. It's the taste of glutamate. This is what we taste in beef and aged cheese. There is a misconception that we have localized taste buds, but we don't. Taste buds are evenly distributed across the tongue, so there is no localization of any specific taste. For taste to be perceived, the chemicals must dissolve in the saliva. The gustatory hairs are then stimulated. We do have different thresholds of stimulation for different tastes. Bitter is the most sensitive, and this makes sense because most toxic things that are bad for us, have a bitter taste to them. The fibers of the glossopharyngeal, facial nerve, and vagus nerve are the ones that are stimulated. These are all of the cranial nerves that are involved in taste. The gustatory cortex is stimulated. This is located in the insula of the brain. The fibers do go to the medulla, going close to the emetic center. This can stimulate vomiting, and to the thalamus, and the thalamus will associate feeling with taste. Since taste is 80% smell, taste is mostly affected by damage to smell receptors. Anosmias are a loss of smell. This can happen as the result of a head injury, a tear in the olfactory nerves. Nasal inflammation may block air from getting to the olfactory epithelium, and with aging, we simply don't replace as fast as we used to. Uncinate fits are olfactory hallucinations. Usually they are unpleasant smells. These can result from brain surgery or head trauma. Some epileptics have an uncinate fit as an olfactory aura prior to a seizure. The loss of taste is rare because there are multiple nerves involved in your sense of taste. Loss of taste is usually associated with loss of smell. The ear is our organ for hearing and balance. There are three major areas to the ear. The external or outer ear is involved only in hearing. The middle ear or the tympanic cavity is also only involved in hearing. But the internal or inner ear is involved in both hearing and equilibrium. There are separate receptors for hearing and balance that respond to separate stimuli. These are activated independently. The outer ear has the auricle or pinna, the lobe here, and the external acoustic meatus. The middle ear starts at the tympanum, has the three ear ossicles, the malleus, the incus, and the stapes, and is connected to the nasopharynx by the pharyngotympanic or auditory tube. And then the inner ear is carved out of the temporal bone. The external ear has the auricle or the pinna. This helps collect and direct sound waves into the auditory canal. The external auditory canal or meatus directs the sound down to the eardrum. There are ceruminous glands here that make earwax. Earwax primarily acts as acoustical tiling to help dampen sound and also traps dust and other things that might irritate the eardrum. The tympanum or eardrum is the boundary between the outer ear and the middle ear. This will vibrate to transmit sounds to the middle ear bones. In the middle ear, we can see the oval window, which is the entrance to the inner ear. This is where the stapes is going to push on the inner ear. The round window is a little safety valve for extra loud sounds. And the ear ossicles, the malleus, the incus, and the stapes, will transmit vibrations through the air cavity that is the middle ear. The pharyngotympanic tube or the eustachian tube helps connect the nasopharynx to the middle ear. This is used to equalize pressure on the eardrum. Swallowing or yawning opens up the eustachian tube, and this helps equalize pressure on the tympanum. That's why you sometimes have that need to swallow or yawn when you're going up in the airplane. When the tympanum pressure equalizes, you hear that little pop in your ears. Because this is also a connection to the nasopharynx, any infection from the nasopharynx may get up into the middle ear. This is when you have otitis media. Fluid can collect in the middle ear, and that can put pressure on the eardrum. If enough pressure is exerted, the eardrum may rupture. A ruptured eardrum may or may not heal, but if it heals, there's going to be scar tissue and it won't vibrate the same way, so there will be some hearing loss. A myringotomy is a small opening made in the eardrum to either drain fluid or to put in tubes so that fluid can be constantly drained. The inner ear is a bony labyrinth that's carved out of the temporal bone. The vestibule is the entrance to the inner ear. Then we have the cochlea, which is where the organ for hearing is housed, and the semicircular canals, which are involved in rotational balance. The bony labyrinth is lined with membranes. This is known as the membranous labyrinth. These are continuous sacs and ducts within the bony labyrinth that are surrounded by perilymph. The inside of these sacs contains endolymph. The fluids will conduct sound, and also fluid motion will be used when we are detecting balance. The cochlea contains the organ of Corti. This is the structure that is involved in hearing. The cochlea is a long coiled tube. It looks like a snail. It's filled with both perilymph and endolymph. There is a basilar membrane that supports hair cells, and the hair cells are covered by a tectorial membrane. The cochlea, this snail-like structure, when it's cut apart, we can see all of the tubes are wound around together. We have these two tubes. These are filled with perilymph, and this inner tube is the one that's filled with endolymph, and this is where the organ of Corti is located. Here you see the organ of Corti. Here are the hair cells. Notice how they are attached to the branches of the nerves. We have some supporting cells. The basilar membrane will have perilymph underneath it, and here is the tectorial membrane that comes across and covers those hair cells. Hearing occurs because sound is collected by the pinna and travels down the auditory canal to vibrate the tympanum. The vibrating tympanum pushes on the ear ossicles, and this carries the vibration through the middle ear. When the stapes pushes on the oval window, it sets up wave action in the perilymph of the cochlea. The waves in the perilymph will move the basilar membrane. As the basilar membrane undulates, the hair cells will brush against the tectorial membrane. This will generate a nerve impulse on the cochlear nerve, cranial nerve 8, and this will be transmitted to the temporal lobe. Auditory processing is a very complex process. The height and frequency of the waves in the perilymph will depend on the loudness and frequency of the sound. When the stapes pushes on the oval window, we set up wave action in the perilymph of the scala tympani. This causes the basilar membrane to move up and down, and the hair cells will then strike the tectorial membrane. When the hair cells bend, this will generate a nerve impulse that will be carried to the temporal lobe of the brain for auditory processing. There are two types of deafness. Conduction deafness occurs if there is any blockage of sound conduction to the inner ear. This can be impacted earwax in the external auditory canal, a perforated eardrum, otitis media with fluid keeping the bones from vibrating, or otosclerosis, which is a fusion of the ossicles. Most of these conditions can be corrected relatively easily. Sensory neural deafness is when there is damage to any of the neural structures. This could be at any point from the cochlear hair cells to the auditory cortical cells. Sensory neural deafness typically results from a gradual loss of hair cells. Loud noises contribute to this loss. Tinnitus is when there is a ringing or clicking sound in the ears when there is no auditory stimuli. This can be due to cochlear nerve damage or inflammation of the middle or inner ears. Some drugs can damage the cochlear nerves. One of the reasons we worry about aspirin overdoses in children is because of damage to the cochlear nerve. Meniere's syndrome is a labyrinth disorder. It affects both the cochlea and the semicircular canals. It causes vertigo, nausea, and vomiting, as well as tinnitus. Meniere's syndrome is not clearly understood. Sometimes it comes and goes. Sometimes you have it for long periods of time. The only treatment is to treat the symptoms. The other function of the ear is equilibrium and orientation. This is a function of the vestibular apparatus. The vestibule contains the saccule and utricle, and then we have the semicircular canals. Static equilibrium is your equilibrium of linear motion, going forward, backward, side to side, or up and down. This is a function primarily of the saccule and the utricle. Dynamic equilibrium is your rotational movement. This is primarily a function of the semicircular canals. The vestibule with the saccule and utricle, and the semicircular canals. These are the parts of the inner ear that are involved in balance. The saccule and utricle, which are responsible for static equilibrium, have areas known as maculae. These are areas of sensory receptors. The sensory receptors, again, are hair cells. They're covered with a gelatinous membrane called the otolithic membrane, and there are calcium carbonate crystals embedded in this membrane. As the head changes position, so do the calcium carbonate crystals. This puts different pressure on different hair cells. This can be interpreted in your brain as forward, backward, side-to-side motion, or even up and down movement. The saccule and utricle are found in the vestibule. The macula have the hair cells with nerves going to each hair cell, the otolithic membrane, and the otoliths, those calcium carbonate crystals. As the head changes position, the pressure of these crystals change, and that's going to bend different hair cells. With the head in an upright position, gravity pushes on the crystals. As you move your head forward, we have different forces that cause different hair cells to bend. Dynamic equilibrium involves the perception of your rotational movement. The semicircular canals are filled with endolymph. At the base of each canal is a swelling known as an ampulla. The ampulla contains a cupula. The cupula is gelatinous material that has hair cells embedded in it. Rotational movement will set up fluid movement in the canals. As the fluid flows past a cupula, it sways. This means that the hair cells are bent. This information is sent to the brain and interpreted depending upon which hair cells are bent. The semicircular canals have the ampulla, the cupula, the gelatinous material with hair cells embedded in it. When you are at rest, the fluid is not flowing. The cupula is upright. We have no bent hair cells. As you begin to spin, fluid will flow, bending the cupula. When you stop, for a while the fluid continues in one direction and then will reverse its flow. All of this information is transmitted to the brain and lets your brain know that you were spinning and now you're not spinning. Nystagmus is a side-to-side movement of the eye. This is in response to rotation. As rotation begins, your eyes will drift in the direction opposite to the rotation. Then the central nervous system will begin to compensate. You will have a rapid jump of the eye toward the direction of rotation. When rotation ends, the eyes continue in the direction of the spin and then jerk rapidly back in the opposite direction and then back in the direction of the spin, and you have this little vibration until everything kind of settles down in the semicircular canals. Motion sickness occurs when you get conflicting information from your visual sources and your vestibular sources. When you're on a boat, you're not moving. There is no vestibular stimulation that you're moving, but your eyes see waves moving or they see things moving past the boat. So now your eyes are sending one piece of information and your vestibular system is sending something else. Your brain gets confused. These signals running into the brain go in close proximity to the vomit center in the medulla. This is what causes the nausea. Anti-motion drugs simply depress the vestibular input. The sense of taste and smell tends to be sharper in the young than in the old. Infants like bland food because they can get a lot of flavor out of that. As the elderly begin to lose their sense of taste and smell, they may eat less. The sense of taste and smell begin to decline in the 40s, probably due to slower regeneration of the receptors. Infants are hyperopic when they're born. They have short eyeballs and they only see gray tones at first. The lacrimal glands begin functioning at about 2 weeks. By 3 years of age, depth perception and color vision are well developed, and they have normal vision by age 6. Presbyopia is a loss of the inability to focus on nearby things due to loss of lens elasticity. This occurs in about the mid-40s. This is when people start holding things very far away from them and have to have readers in order to read fine print. Newborns will have reflexive movements to sound. By the age of 4 months, they begin to tune into family voices. Toddlers begin critical hearing with the developing of vocabulary. They begin to try to imitate the sounds they're hearing. Presbycusis is old hearing. This is when you begin to lose your hearing as you age. Primarily, the hair receptor cells wear down. The hairs get shorter. This means we need a louder sound to trigger an impulse. High frequency ranges are generally worn down first.